All right, yeah, I think we can get started, and I think we'll have a couple more people rolling in in the first couple minutes. But yeah, thank you everybody for joining today. And yeah, please introduce yourselves in the chat, get to know each other. Um, and yeah, welcome to our last Science of Alt Protein APAC edition for 2023. And yeah, thanks to all of those who are joining us new today or who have been attending earlier this year as well. My name is Arun Naidu, the SciTech Engagement Specialist at GFI APAC, and I will be your host for today. And it is with great pleasure that I welcome Dr. Iris Haberkorn as our speaker to talk to us today about nutritious single-cell-based foods for more sustainable and healthy diets in urban areas. Dr. Iris Haberkorn is currently the project lead and senior scientist at ETH Singapore Centre, leading the urban microalgae protein project. She has a background in nutrition and food science from the University of Hohenheim, Germany, and ETH Zurich, Switzerland. She did her PhD at ETH Zurich in sustainable food processing under the supervision of Professor Alexander Matisse. Her current work at ETH Singapore Centre focuses on establishing solutions for urban food production and processing based on microalgae to establish more resilient, sustainable food systems, harnessing circular bioeconomy concepts. Today's talk specifically will highlight the potential of single-cell-based value chains with a focus on microalgae as a dense source of micronutrients for resilient, more sustainable food production in highly urbanized areas. Microalgae are considered the most resilient and dense source of micronutrients. The consumption of 2 to 10 grams of microalgae could provide most of the micronutrients required in a healthy diet. However, the bioavailability and bioaccessibility of microalgal nutrients is limited, owing intricate cell structures. The talk will address the role of emerging processing, harnessing innovative biological and technological approaches, such as the establishment of pulse electric field-based biorefinery to improve nutrition, leverage efficiency, and consequently foster the application of nutritious, affordable microalgae-based products on the market. Yeah, we're super excited to hear your talk. Before I hand it over to Dr. Haberkorn, I would like to make one quick announcement. Today's seminar will run for about an hour with a total of 15 minutes at the end for Q&A. On the bottom of the screen, you'll see a chat and Q&A button. We encourage you to use the chat function to introduce yourselves to other participants. However, please make sure to direct your questions for Dr. Haberkorn to the Q&A window so that we can address them easily. In the Q&A section, you can also have the option to upvote questions you would like to be answered, and you can prioritize those that are most relevant to what you're interested in. So yeah, please, as we go through the talk, feel free to add in, and then I will moderate the Q&A session at the end. But yeah, with that, thank you so much. And yeah, please take it away, Dr. Haberkorn. Thank you.
Okay, great. Thank you so much, Arian, for the very kind introduction. And I also would like to say a word of thank you. Firstly, of course, to the GFI for the invitation to host this webinar, and everyone, of course, for attending and showing interest in the research that we are doing here at the Singapore-ETH Centre, also related to ETH Zurich, like Arian mentioned, the institute that I come from, and that I was doing my PhD. And I'm looking forward to a great discussion in the end. But first, I want to give you an introduction of what we're actually doing here at Singapore-ETH Centre at ETH Zurich and the rationale behind what we are doing. And I was asked today to give a more technical talk. So I will try to also provide you some kind of the basics or the fundamentals behind the research that we are doing. But first, to give you some background of the rationale or the reasons why we are conducting the research. When I was discussing with Aaron before, I heard or I learned that most people that are listening today are from the Singapore ecosystem. And you all should be aware that especially Singapore is very keen on pushing self-sufficiency in food supply. Because currently Singapore is importing 90% of its nutritional demands, and by 2030 they aim to leverage this to 30%. However, arable land in Singapore is highly limited. We are only at 1% arable land. So we need to find innovative solutions that allow us to produce food here independent of arable land. And of course, this not only translates to Singapore, but it's also immensely important to find novel, sustainable, more resilient food systems to transform our food system. And this is reflected partially in some publications that you see here on the left on the slide that we were part of, like for the planetary health, the ETH Lancet publication on nature sustainability. So in research already since a couple of years, there's a huge focus on shifting our food system, providing data and insights. And luckily, what I learned at the weekend is now that also at the COP28, food systems now also played a major role, which is very nice for us to see. Although I also learned that key target actions were not defined, but it's amazing to see that future and food system transformation in particular is now on the agenda of global climate change, also for policy and decision makers. And we hope that research and the data that we are providing at ETH, at Singapore-ETH Centre, and the ecosystem, for instance, that we have in Singapore here can foster this change and bring transformative change to improve the sustainability of our planet and to not transgress planetary boundaries anymore. So the challenge that we are actually facing is that currently also the main focus of the COP28, largely promoted by Johan Rockström, we are transgressing the planetary boundaries, and we see that our food system is actually a major contributor to anthropogenic climate change. We are causing a third of the global greenhouse gas emissions. Also, we are using the majority of the arable land that is available for food production. And it's estimated that more and more people will be living in urbanized environments, also putting pressure to developing solutions that are applicable for highly urbanized environments and that are independent of arable land. So, and we see that already now we are transgressing the planetary boundaries with the systems that we have, but until 2050, with the growing population that we are facing, we would need to increase food production by around 60%. However, a third of the food that we already produce, although we are using large resources already, is wasted along the value chain. So we also need to find solution on how to mitigate or to, as a first priority, avoid food waste. So we see that a lot is actually wasted, but already now a lot of people are suffering from insufficient food supply. So we really need to find more sustainable solutions that are aligned with the sustainable development goals that will allow us to feed a growing population and that will stay or allows us to stay within the planetary boundaries. And our group at ETH Zurich is focusing on developing such solutions, and the research approach and the topics that we are discovering at our ETH Sustainable Food Processing Laboratory, you can see on the slide displayed here. So you see on the bottom part that actually our research and the key research actions that we are, or target actions that we are identifying is based on multi-indicator sustainability assessment. And here we do 3-dimensional multi-indicator sustainability assessment, which means that we also factor in the health and the nutrition domain, which is often not done in a typical life cycle assessment, but we also need to characterize the nutritional and the health parts to really identify whether the new solutions that we develop, for instance, alternative protein solutions, are really more sustainable in terms of economy, in terms of planetary health, but also in terms of nutrition and human health. So we identify our key target actions based on 3-dimensional multi-indicator sustainability assessment, and our research focuses actually on covering the whole value chain, which means that we go from raw material production, focusing on microalgae, but also black soldier fly larvae. We go over processing, disintegration, extracting components, characterizing components like proteins or lipids, for instance, from microalgae. We process them in alternative protein-rich foods with enhanced nutrition. And then we also now step more and more into the nutrition or medical domain to assess whether our products, our raw materials actually have the nutrition that they should be developing, not only from a protein profile point of view, but really from a nutrition, meaning bioavailability, bioaccessibility point of view. And here the black soldier fly larvae, I should mention that we are not focusing on an application of black soldier fly larvae for food, but for feed application, for instance, more sustainable aquaculture feed. And I think aquaculture is also a big topic here in Singapore with urban vertical farming for I don't know, for shrimps or for fish. And here black soldier fly larvae that we are rearing on organic food streams, waste streams from urban environments could be a very promising solutions to tackle food waste in cities like Singapore. And I already mentioned to you that we are using multi-indicator sustainability assessment to identify environmental, economic, and social impacts. If you're interested in the different domains that we are covering, I just wanted to show the publication, and you see a table from the publication displayed here on the list. So if you're interested in that and certain key target actions that we identified, you can check that publication. And here we were looking at different metrics, for instance, food nutrient adequacy, ecosystem stability, but also typical indicators or metrics like socio-cultural well-being. However, what is a clear trend that we can see from these multi-indicator sustainability assessments is that due to high environmental impacts, we need to reduce animal-based protein-rich foods. So meat production for land use change, but also for methane production. But what we currently see is that alternative protein-rich foods often do not deliver the nutrition or nutritional profiles that meat, for instance, would be delivering. Sometimes alternative products have adequate amino acid profiles, but in very often or in many cases, they don't. So, and then some companies or some research is looking at delivering adequate amino acid profiles, but it's not looking at the bioavailability of the proteins. And then also if we compare it to meat, for instance, we also need to look at the micronutrients and the bioavailability, bioaccessibility of the micronutrients that the meat would be bringing as a daily dietary components, like for instance, iron, but also vitamin B12, zinc. Where we currently face huge micronutrient deficiencies all around the globe. So in case we are looking at developing novel solutions, alternative protein-rich solutions, we need innovative food nutrition technologies for more sustainable and affordable protein-rich foods, but also at solutions, technologies that deliver adequate nutrient profiles, looking at proteins, available protein, but also at micronutrients. And what we are doing currently here in Singapore is that we are touching upon that research. We are developing alternative protein-rich foods with a focus on fish analogs, meat analogs, using heterotrophic microalgae. But here a clear focus for us is also the application focus, because Singapore is more towards implementation and less on the very basic research side, but we really want to see applications on products, on solutions that we can transform the system here in Singapore and that can be used also by industry. So what we are doing here in Singapore is that we are developing urban food production and urban food processing context. And for the urban food production, we are, like I already mentioned, using heterotrophic microalgae, so microalgae that are growing independent of arable light, of light, and that can just be grown, for instance, in void spaces. And the nice thing of heterotrophic microalgae is that they're using organic carbon as a source for carbon. And here we are not using glucose, but we are trying to replace common media that are used in industry by food industry side streams to also establish circular bioeconomy approaches. And here we are using side streams from tofu production like soy whey, but also brewer's spent grain to grow the microalgae on. And the preliminary results from our research partners look very promising, actually, because we could see that we can entirely replace conventional media by using these food industry side streams to grow the microalgae upon. And we have the same biomass yields and productivities as when using industrially commercialized media. And what ETH or Singapore-ETH Centre is focusing on is developing pulse electric field-based biorefinery solutions to leverage microalgae growth for reasons that I will explain later. And we also use it for extracting certain components from the microalgae or making them better bioavailable. We then use these components. First, we use the entire microalgae because of their holistic and inherent nutrient profile to include the whole microalgae in fish analogs that we are producing with 3D printing, but also with extrusion to have more centralized and decentralized options. We are also working with only isolated protein from the microalgae, and we couple this with consumer-centric evaluation to really identify what the consumer, and especially the Singaporean consumer in that case, wants or expects from plant-based meat analogs or fish analogs, because often we see a fail on the market because we are producing products that actually don't fulfill what the consumer is expecting, and then we are not hitting the price targets in addition. So yeah, and what we want to avoid is really a decline in interest and a decline in trust of consumers in these alternative protein products, because they can have such a huge impact on transforming our food system. And here we partner with different companies like Nestle, Bühler, Givaudan, and also the Singapore Food Agency. Why are we focused on microalgae? Because for us, they are not only an alternative protein source. Here on the picture, you can see phototrophic microalgae, because during my PhD, I was still working with the phototrophic microalgae, and they still make a really, really nice picture. But of course, if they were heterotrophic, they wouldn't be green, they would be yellow, which we assume is better perceived by the consumer, because if you use green microalgae and you try to process them, they might turn grayish or brownish, and then these products don't look very appealing. So microalgae are not only an attractive protein source, but they also have amino acid profiles that are in line with WHO and FAO recommendations. They have other nutrients like essential omega-3 fatty acids like EPA and DHA. They can grow independent of arable land, and they have way higher biomass productivity than conventional crops that are applied, like soy or pea protein, for instance. And one point that is very attractive about the microalgae, especially if we look at protein, is the low land requirement that we have for microalgae for protein production. Here you can see a comparison of different conventional protein resources displayed, and the actual land demand that we have for these protein resources per kilogram of protein. And you can see that even plant-based protein has slightly higher land demand than the microalgae. And for the microalgae, we are very, very low in the land that we require, with 1.6 square meter per kilogram of protein, which of course is very attractive for urban environments like Singapore. But why are microalgae now, from a nutritional point of view, also very interesting for us? Because they are they have high production efficiencies, and they have bioaccessible protein. So on the slide displayed here, you can see actually that we are, since some months now, working not only with conventional microalgae like Chlorella vulgaris, but we are working more also with extremophilic species like Galdiera sulfuraria, which you can see displayed here on the left. It was actually discovered in volcanoes, on the crater of a volcano. So some crazy scientist must have gone to a crater in a volcano, climbed in there, and discovered that a high potential microalgae is growing in there. But this Galdiera sulfuraria is actually really, really promising for us, because it can grow at pH 1, so you don't have any issue with contamination, and it can grow at temperatures of 42 degrees Celsius. So here with these conditions, low pH, for instance, we don't have the problem of other conventional microalgae cultivations of contamination. So we can just grow them without any measures of, I don't know, sterilization of probes or anything else of media. And what we also see is actually that the biomass productivity is extremely high for Galdiera sulfuraria. That is 2 2.3 times higher than for autotrophic cultures, for instance. What was very interesting for us to see, if you now look at the protein, but also the nutritional point of view that I was mentioning before, from bioaccessibility, so not only protein content, but really how much can our body absorb and use of the protein that we are delivering. And if we were comparing two different Galdera sulfuraria species, we saw that actually both species have different protein contents. So one had 64% protein, the other 51% protein. But if you then look at protein bioaccessibility, is actually that the species with a lower protein content had a higher bioaccessibility. So this showed to us that sometimes we cannot only look at the total protein content of microalgae, but we also need to see and then decide on the species, the cultivation conditions, how good the uptake of the protein, so the bioaccessibility actually is. And that one point could be for Galdera sulfuraria, for instance, that it doesn't have such a harsh cell wall as other microalgae species like Chlorella vulgaris or Auxenochlorella would have. What we then make use, or what we then make from the microalgae, depends on the application. So depending on the source that you can see here on the left side, we have a lot of different components in the microalgae: lipids, vitamins, proteins, minerals, and we can use the whole microalgae or only the protein that we extract if we look at a macro scale for extrusion for plant-based meat analogs. But we can also disrupt the microalgae, use them for the stabilization of foams of or emulsions, for instance. Or if we go very much on micro scale, we can only extract certain components like a high value phycocyanins or pigments. Phycocyanin being the only natural blue pigment available on the market that is used commercially by companies like Nestle for Smarties production. And then the species, but also the processing depends on the final application, but also, of course, on the price point. And what we were investigating at ETH Zurich was different applications on how to create functionality through microalgae protein. Here mentioning the high moisture extrusion, where we were able to produce the first microalgae-based meat analog. But also for emulsions to, in the end, deliver nutritional value. And if you look at the extrusion part, we were using different microalgae contents, different moisture contents, and we obtained, as you see on the picture here, different fibrillar structures from not very brittle to nicely fibrous to very brittle. So what was very attractive for us to see was then, if we look at the nutrition point of view from microalgae, that microalgae could not only deliver nice structure when combining with soy protein in that case, but that we could also retain the nutritional values of the microalgae. So these were not destroyed, or to a large extent not destroyed, like if you look at vitamin B2, for instance, and retained in the exudates. This shows us that actually microalgae could be a valuable supplement to or addition to plant-based meat analogs if we look at natural ways of providing nutrition to alternative protein-rich products. But in this study, we only looked at the nutrient content of the microalgae analogs, which means a pure chemical or food chemical analysis, right? But not at the bioavailability. So the next step would be then also look on how much of the protein that we actually deliver in these plant-based meat analogs would be bioavailable, bioaccessible, but also the nutrients. Because these microalgae like Auxenochlorella prototrichoides, like I mentioned before, they have a very harsh cell wall. So in this case, it would be important to do some kind of processing that we will touch upon later with the PEF research that we are doing to make the nutrients also bioavailable and not just include them so they're not somehow digested or taken up in the end. If we then go, so this is a more macroscopic point of view. If we then go more into microscale, we are also looking on isolating and purifying protein fractions, in this case from Arthrospira platensis, so spirulina, which is has a very nice spiral form that you can see here on the left. And that we were looking at the microalgae protein, and we compared it to conventional, so the techno-functional properties we compared to conventional animal-based protein for network formation. And this, by the way, is then also important if you want to apply it in extrudates, plant-based meat analogs, because so far, actually, the mechanism on how these structures in plant processing during extrusion are formed is not entirely clear. And here it's also important to understand the underlying techno-functional properties of the proteins that we're extracting from the microalgae. And what we could see for Arthrospira is that with the extraction methods that we were using, that we obtained, so we just did filtration and protein purification via diafiltration. We saw that the protein yield for isolates compared to the crude extract for Arthrospira platensis was, so the protein yield was lower, of course, the purity was higher. There were some differences in the layer thickness, but for pure protein isolate, if compared to just crude extract from microalgae, we saw actually enhanced functionality. So then in the end, the decision is actually on the application and the cost point on whether we want to produce microalgae crude extracts or protein isolates for replacing animal products or animal-based food ingredients for stabilizing interfaces. Then if we look at microalgae, very often a point that we are discussing, and what is also a reason why we are conducting the research here at Singapore-ETH Centre, is a lot of people talk about microalgae as a more sustainable or sustainable solutions for producing protein. But what we currently see is actually that if we compare it to traditional benchmark proteins, microalgae are not yet more sustainable compared to what we also need to say, highly optimized systems like soy meal or chicken production, and then or chicken-based protein production, which you can see here in the environmental life cycle sustainability assessment that was conducted also as part of the research that my professor Alexander Mattis is doing. And here they were comparing different benchmark alternative protein sources, for instance, chicken, dairy, gluten-based, insect-based. You see still huge environmental impact from lab-grown meat with very optimistic scenarios, but the data is also from 2015. So there might have been some changes here in the energy consumption, for instance, that is associated with lab-grown meat. But this is also one reason for us to focus on heterotrophic microalgae, as the environmental impact that we see associated with microalgae depends hugely on the cultivation mode. And you see here that actually heterotrophic microalgae production is way more eco-efficient if you compare it to phototrophic microalgae production. But still, we need to do more technological improvements, improve the technology readiness level, but also economy of scale for microalgae production if we want to make it more energy efficient and more really a more sustainable, attractive solution as alternative protein for future food applications. And here you see a comparison of the environmental sustainability where nutritional impacts are not included of most relevant protein resources. On the left, you can see soybean meal, rapeseed cake. Here, HM is actually for insect protein, but also on the bottom part, the microalgae. And what we actually see is that the energy demand for microalgae production is still very, very high. And here you see two different numbers on the bottom. So a range between 217 and 4,181 megajoules. And the 217 megajoules are associated with heterotrophic microalgae production, and a large number of 4,100 with phototrophic microalgae production. But if we compare it to soybean meal or protein that we extract from soybean meal, actually the energy demand behind microalgae production is still very high. So we need to find solutions to decrease the energy demand that we see associated with microalgae, because from the nutritional and protein point of view, they're an extremely attractive solution, especially for urban environments like Singapore, where arable land is extremely limited. And this is why our research, I was mentioning it before in theme 2 from SEC, Singapore-ETH Centre, is focusing on pulsed electric field-based biorefinery and process innovation, because by applying this pulsed electric field biorefinery, we aim to leverage single cell growth, but also extract proteins in a more energy efficient and mild manner than it is currently done. And here, pulsed electric field processing, to go more into detail into that direction, I think the background in the introduction was also mentioning the pulsed electric field-based biorefinery. The attractive point about pulsed electric field application is that we have multi-product applications, and it's more new currently to the biotech industry, and it's already applied, especially microsecond pulsed electric field processing, for different applications like decontamination, microbial inactivation at very high energy inputs, but for heat sensitive liquids where you want to preserve vitamins and other nutritional components, but also for structure modification, for instance, applied for potato processing to make that process of frying less fat intense. And the pulsed electric field-based treatment mechanisms depend largely on the energy input and the treatment that you are applying. So depending on the specific energy input that you apply, the applications are different. And here we are looking at different, actually, we treat it as 2 different technologies, like microsecond pulsed electric field and nanosecond pulsed electric field, because they're working with 2 different mechanisms, or it's not entirely different, but different processing windows that trigger different responses in the cells. And for the microsecond pulsed electric field, you have more an effect on the cell membrane level, and you can cause irreversible electroporation leading to cell death, but also reversible electroporation that is attractive for introducing molecules into the cells, but also, of course, selective extraction of molecules. And then if you look at nanosecond pulsed electric fields, we actually saw that one can enhance cell proliferation in cells by inducing intracellular sublethal stress. And as a background, the pulsed electric field processing, as I mentioned, depending on the processing window apply, acts either on outer membrane level or on intracellular level. And actually, what you create if you apply an electric field to a cell is a transmembrane potential. Each cell has an inherent transmembrane potential, but also a certain threshold. And if you exceed that threshold of the transmembrane potential, you cause the lipids in the lipid bilayer, so the phospholipids, to flip. And depending on the energy input and the strength of your treatment, in this case, we are talking about microsecond pulsed electric field processing, which is mainly acting on cell membrane level. So depending on the strength of the electric field and the energy input, you're creating reversible pores that can reseal again. So the phospholipids, they can flip back in their original position, and then the cell is sealing again. But you can also cause irreversible electroporation, which is then leading to cell death. And if you look at the bottom right graph here, actually this processing window applies to this green highlighted field that you see here on the left. So this means that if we are operating with an angular frequency below 10 to the power of 7, we are mainly acting by applying a or increasing a certain transmembrane threshold on plasma membrane, cell membrane level. However, if we apply very short pulses in the nanosecond domain that are very strong, so with pulse amplitudes between kilovolts and megavolts, we can actually act, or we actually act more on organelle level, which means that we are triggering intracellular structures and not on plasma membrane level anymore. And this then is associated or connected to inducing intracellular sublethal stress inside the cells, not causing cell death. And here we can see different applications, and I will mainly explain the applications that we are investigating at Singapore-ETH Centre and ETH Zurich on single cells and proteins and lipids. But what I was mentioning before, if we are looking actually at microsecond pulsed electric field, it already finds applications in microbial inactivation, but also on working with plant cells, because you can actually create those pores very gently, and then you can release, for instance, water. So you have a loss in toggle pressure inside the cells, and then you need less frying fat, for instance, for the potatoes, which is one very prominent application. First, looking at the nanosecond pulsed electric field processing, because it comes as a first step in actually the cultivation process. If we look at the pulsed electric field-based biorefinery, we find different applications looking at nanosecond pulsed electric field or microsecond pulsed electric field. And then, of course, if you have a cultivation, what you first would like to do is to apply nanosecond pulsed electric fields to stimulate growth. And to obtain higher biomass yields from your microalgae, and then combine it with microsecond pulsed electric fields to end membrane separation, which you see displayed here on the right side in the graph, at a later stage of the cultivation to really extract the components out of the cells. However, this is mainly now relating to batch cultivations. One next step would be to apply it also to continuous applications, where you can do something that is displayed here on the right top part that is called microsecond-based milking, where you can continue, or where the idea is to continuously extract components from microalgae, other single cells, by inducing these reversible pores inside the cells. Looking at the nanosecond pulsed electric field, so the first step in the cultivation process actually is one crucial thing, that we are enhancing cell proliferation, but it highly depends, the application of the treatment highly depends on the cellular growth stage. So you only achieve growth promotion if you apply the treatment in the early exponential growth phase, so the high proliferating state of the microalgae. If you apply this, we actually saw that for different single cell organisms, so for Arthrospira platensis, for Chlorella vulgaris, and to phototrophic and heterotrophic growth, but also for yeast like Saccharomyces cerevisiae, we can trigger cell growth and cell proliferation by up to let's say 17–20%, depending on the species. However, what we could observe was that we can trigger this response in different organism domains. So not only microalgae, but also bacteria and yeast. And this effect was actually first observed for Arabidopsis seedlings that you can see displayed here on the right. So the treatment was applied to the seedlings, and then germination happened significantly faster if compared to other seedlings that were not treated. And this, of course, would then be interesting if you look at concepts like vertical farming, where you want to probably speed up your seedling growth. So here, nanosecond pulsed electric field processing could also be a promising technology for vertical farming. What we also saw was that in Arthrospira platensis, we could significantly increase phycocyanin content. I was mentioning before, phycocyanin is a high value component and the only blue pigment that is commercially currently used by companies like Nestle in their food production. However, to go more on the technical side again, this graph is very messy, but what it actually should show is that the effects on how nanosecond pulsed electric field processing leverages single cell growth are still largely unknown. And there are different concepts that are discussed in literature, but they are not related to microalgae and not related to growth promotion. But the common hypothesis is that nanosecond pulsed electric field processing triggers the release of calcium from intracellular storage components like vacuoles or mitochondria, which is then resulting in the induction of signaling pathways. And that in a downstream response are then leading to enhanced cell proliferation, but also the generation of reactive oxygen species. So if there's some kind of molecular biologist that would be very keen on working together with us, please shout out. We would be very happy to actually investigate this mechanism to be better able to tailor the response and the treatment to different organism domains. And here, actually for the nanosecond pulse electric field processing in Singapore, we are not only looking at lab scale application, but a clear goal is also to upscale to industrially relevant pilot scale. And here we are working with our partner Bühler, where they developed a scalable system for this nanosecond pulse electric field application, which will allow us to actually treat the microalgae cells not only on lab scale, but also scale up, like I said, to industrially relevant pilot scale. And for the microsecond pulse electric field processing, we then look more into extracting certain components of interest. Here for us, what is interesting to see is that we can actually preserve the techno-functional properties of certain components inside the cells. And here you can see, again, a picture of different application that microsecond pulse electric field is used for. And that is actually, targeting for plant cells to cause a loss of barrier function and then a leakage of intracellular contents. But we have very limited effects on vitamins, flavors, or proteins on the cells. For our application here at Singapore-ETH Centre and ETH Zurich, mainly the cell death part is currently interesting, and that I will be showing to you. What we are here aiming is to develop a more energy efficient way of extracting the proteins by combining the pulse electric field processing and then induction of irreversible pores on cell membrane level with inducing autolysis in the cells. And the theory behind this is that you have a more mild microsecond pulse electric field treatment. You see it displayed in the middle on the graph here on the slide, and you combine it, or you electroporate mildly to create pores, but then this treatment should also induce certain enzymes that are associated with autolysis, and you combine this then with an incubation step that allows us to extract proteins in a more mild way by keeping their techno-functional properties. And we compare it here with high pressure homogenization and sonication to especially high pressure homogenization technology that is commonly applied, but of course applying a lot of energy and destroying the components in their original techno-functional properties, but also causing emulsification and mixing of certain components. So you also need downstream processing and heavy membrane filtration in the end again. However, if you apply microsecond pulse electric field processing and combine it with an incubation step, we actually see low energy consumption, we have mild conditions, and we only partially disrupt the cells. And here I will show you some results that don't relate to the protein, but more to lipids. We were doing a project together with Nestle in Lausanne, where we were using microsecond pulse electric field. And the idea here was to induce autolysis and to weaken the cell membrane, but still keep the cell structure. Because for lipids, one main application actually from microalgae industry to replace fish oil is, so one issue is that we are causing, if we are extracting the lipid from the microalgae, is lipid oxidation. And if we can only partially impair the cell membrane by microsecond pulse electric field processing and autolysis, but somehow keep the cell wall, we have some kind of microencapsulation of the lipids, but then the lipids would be more or better bioavailable because we are impairing the half cell wall of the microalgae. And here actually we were quite successful in showing this. So we combined the pulse electric field treatment with an incubation step that was inducing cell wall degradation by endogenous enzymes. And we could actually see that over the incubation time, the lipid bioaccessibility, so not only availability, but bioaccessibility increased. And here you can see displayed 4 different temperatures. And even at 4 degrees Celsius, we reach the same bioaccessibility, but after longer incubation than at higher temperatures. And we could see here that actually what I was describing before, which also applies to protein, HPH leads to the highest lipid bioaccessibility, but PF or pulsed electric field processing does not impair cell integrity, which of course, like I mentioned, then goes towards a more kind of microencapsulation approach. You increase lipid bioaccessibility, but you prevent lipid oxidation from releasing the lipids, so they are more stable, which could then, for instance, be attractive for using these treated microalgae cells and plant-based meat analogs, because here we can increase the nutritional content of plant-based meat analogs, but also the bioavailability and holistic nutrition of plant-based meat analogs. Yeah, and then one idea for us, of course, is to combine nanosecond pulsed electric field processing with microsecond pulsed electric field processing to apply it in a biorefinery approach. And here on the bottom part, you see the main publications that we were using in case you should be interested in reading more. Yeah. And this is again just summarizing, combining nanosecond PEF with microsecond PEF as biorefinery approach, also for urban applications, because the PEF system itself is really, really small, so a low footprint, land footprint, and it could be applied to urban settings like Singapore, combined, for instance, in the downstream with a membrane separation approach. And yeah, this already leads me to the end of the presentation. I hope I could give you some valuable insights behind the motivations of our research, what we are in generally doing at ETH Zurich, but also Singapore-ETH Centre, and then in more detail why microalgae are attractive and why we are using, for instance, why we are, for instance, working on pulsed electric field processing to leverage microalgae, make them more attractive, but also leverage them as holistic kind of nutrient platform. Yeah, thanks of course to all the students who do all the hard work that you see here with the group displayed at ETH, our partners. Yeah, and if you have questions, just feel free to reach out to me and to ask your questions. Thanks so much for your attention.
Thank you very much, Dr. Haberkorn. That was really insightful, and I think super interesting to get the deep dives into heterotrophic microalgae cultivation, as well as the pulsed electric field based process. And yeah, all of the functionalities or outcomes you can get from using that. So yeah, I really appreciate it very much. I know I have a bunch of questions, but it looks like we have questions coming in to the chat already, so we'll get started with that. Yeah, for our first question is there pre-treatment available to increase the bioavailability of micro algae proteins? I think you addressed this a little bit, but
yeah. So I just showed it for the lipids, right? So we didn't address it for the proteins yet. And thanks for the question. I think it's a very good question, and something that we start exploring more and more. So for the lipids, we saw that the pulsed electric field processing can be a valuable pre-treatment to trigger partial disintegration of the cell wall and thereby increase bioavailability. And what we are currently investigating is also to apply this for proteins. And as a next step, we are currently at ETH building up a nutrition platform together with the nutrition group, nutrition and epigenetics, to also investigate micronutrients. So if we apply these pre-treatments, or for instance, not only pulsed electric field processing, but changing cultivation conditions, for instance, whether we can increase the bioavailability not only of lipids, but also of proteins or micronutrients like iron or zinc or calcium.
Yeah, thank you so much. I think that definitely helps clarify on that point. And our next question we have, saying, may I know what is the harvesting technique applied to harvest microalgae biomass prior to extraction and processing, and how to leverage the high costs associated with chemical or organic nutrients for microalgae cultivation?
Yeah, so thanks a lot for the question. I think also two very relevant questions, and I think it's two parts. So the first is the downstream processing, and the first is how to optimize the cost. I will first go on the cost because I think it's a shorter answer. So actually, I got the manuscript from our partners at NUS yesterday, and we have very promising results, for instance, on using food industry side streams that we can leverage. So I mentioned it in the presentation that we can achieve the same biomass yields, but actually our costs for the nutrients for the microalgae are 93% lower because we are using food industry side streams. So what we then use is, okay, of course, this is only the raw material costs, and currently capex or energy costs are not factored in. But what we see is that by using food industry side streams and by hydrolyzing them, what we are currently doing, we can reduce the costs for the raw materials by 93% for the cultivation. And then for the downstream processing, the harvesting hugely depends on the species that you have. So if you have very high concentrated species that grow at, I don't know, 70 grams per liter, 100 grams per liter, actually there's not so much more water compared to, for instance, phototrophic cultures where you only work with 2 grams per liter. So here what people would do is, for instance, work with centrifugation to just centrifuge, use gravity, and then remove the water. And then some people also work with filtration, but I think centrifugation is commonly used for downstream processing prior to extraction. But we try to not decrease water content and get rid of water content, but extract the components with PEF in the water in the system, and then use membrane filtration to separate the components. So we circumvent this concentration step and the energy associated with it.
So I guess it will vary a bit depending on the strain of microalgae.
Yeah, and the biomass concentration that you have, because depending on the biomass concentration, of course, you have more or less water, and then you need to take more or less measures. Also, it depends on how fragile your microalgae are. So some are really robust, and you can apply, I don't know, harsh centrifugation conditions, but other species, they would be less robust, and you need to be more careful if you don't want to destroy them.
And just one quick reminder for the audience too, like the food waste streams you're using, or you mentioned from tofu industry, from spent grains. What else are you using?
So probably it's also important to mention why we're using food industry side streams and not other side streams, because we consider them as more safe than, for instance, using other waste streams. They should, in theory, be free of antibiotics or drug residues. If we use them from food industry side food processing. And here in Singapore, we were focusing on, like I said, brewer spent grain side streams from tofu production, because they are the main side streams that we find in Asia, actually, but also from coffee grinding, all those solids. So coffee was, I think, working not so good, but if we combine soy whey and brewer spent grain, we actually get the nutrients that we require. And then other groups would, for instance, looking more into using dairy side streams. I think this is also very promising. Other side streams can also be promising, but of course, then we also need to look again at the approach on how to cleave the components inside the side streams. If you look at brewer spent grain, we have cellulose, lignocellulose, which cannot readily be used by every microalgae species. So you need to apply some kind of treatment to cleave it into monosaccharides like glucose or fructose, depending on your species. But in theory, a variety of side streams can be used, but for our cultivation mode, they need available sugars like glucose or fructose.
Our next question is, algae are a good source of protein and bioactive components, but they also compose of various toxins at different levels. My question is it possible to utilize harmful algal bloom after detoxification in the development of food nutraceuticals? And can you also give some idea of nanobionic approach in the microalgae culturing? I'll also just add another question related to this was, are there any specific regulatory controls on microalgae based foods? Yeah, maybe 2 questions combined.
Yeah, very interesting approach, actually, and a very controversial discussion. So if we go about I really understand the question, to be honest, because we see that actually microalgae blooms, right, they grow like crazy. In these species, they are in very harsh conditions. They actually shouldn't be there, and then they're just blooming like crazy. And here we are working with highly optimized systems, but we are struggling with obtaining yields and the growth that we actually would need, right? So it seems very attractive to use microalgae species from algal blooms. However, for food applications, of course, they need to be clarified as safe. And for us, we are only working with microalgae species that have GRAS status, so that are recognized by FDA and Food Safety Authority. Of course, very promising, or I don't know, interesting to use species from algal blooms. But for food applications, I'm not so sure. I don't want to, I don't know, harm people. So we would try to stay with the ones that are recognized as safe and thereby approved, and they underwent a lot of investigations to prove that they are safe. And then for the application in food, so there are species that are recognized as safe. Some have novel food approval that have undergone approval. I think Gardera sulfuraria is currently in the process. Looking here at Singapore, we are currently discussing with Singapore Food Agency, and it looks good that we can use our microalgae because it's recognized as safe for food applications. And in Europe, they have slightly different regulations, but certain microalgae species are already used for food applications here. And then what was it?
Nanobionic approach in microalgae.
I unfortunately don't. I'm not aware, so I cannot comment on that.
No, but yeah, thank you so much for addressing the other parts of the question. We have a lot more questions, but I think maybe we have room for just one more for within this hour. And Dr. Haberkorn's email will be sent out, so please feel free to email her your other questions as well. But our next one is, have you done POC on microalgae biorefinery process to maximize the value chain of the microalgae fermentation system? For example, the end product is the protein, the byproducts are dyes, bioactives, etc., etc.
Can you repeat the first part of the question?
Yeah. So have you done a proof of concept or POC on the microalgae biorefinery process to maximize the value chain of the microalgae fermentation system? Yeah.
So a POC, so far for the Singapore project, no, but it's part of the project at the end now, because there's also a requirement, of course, from Singapore, because it needs to have a clear application focus and applicable here. But what we are doing, and what will also be our next step, is to also combine it with life cycle sustainability assessment to see whether the innovations and the technologies that we are suggesting here, like PEF, membrane filtration, extrusion, are actually more sustainable, and then also factoring in the nutrition part. But here we also need to do nutritional assessments to see on the nutrient quality, nutrient content, bioavailability of what we are producing. But definitely it's on the to-do list for the next year.
Yeah. No, yeah, that makes a lot of sense. But yeah, thank you so much. Clearly this is a very exciting, insightful talk for everybody. And we really appreciate your time today and for addressing the questions. It's now 11, so I will close off this session. And yeah, thank you everybody for attending as well. And we look forward to you all joining us next year for our next series of talks as well. Thank you again.
Thank you so much, everyone.
Yeah, it's really great.